Welcome to computational genome biology. In the late 1970s, Sanger sequencing allowed us to read the human genome for the first time, but the process was slow, reading DNA fragments one letter at a time, which made whole genome analysis an incredibly resource-intensive task. That changed in the mid-2000s with the introduction of massively parallel sequencing, widely known as next-generation sequencing, or NGS. Instead of processing a single fragment, modern platforms read millions of fragments simultaneously. Whole genomes can now be sequenced in days, with a dropping cost that makes large-scale genomics accessible to research labs worldwide. But this speed creates a new problem, a massive data deluge. A single modern sequencing run generates terabytes of raw, unstructured, and noisy data. Billions of DNA letters are useless on their own. We need a massive computational engine to filter that noise, align the fragments, and translate the code into specific medical insights. This infographic outlines the 7-step bioinformatics pipeline, the engine that processes raw DNA fragments into readable data. It starts at the bottom with quality control. Algorithms like FastQC evaluate the raw sequencing data to identify baseline errors, sequencing biases, or low-quality reads. Next is preprocessing. Tools such as Trimomatic physically clean the dataset by snipping away artificial chemical adapters and filtering out any remaining low-quality bases. Step 3 is read alignment. At this stage, short DNA sequence strings are mapped against the known reference human genome using tools like BWA or Bowtie. This reference-based alignment is crucial because it aligns the patient sample against standard human DNA, creating a reliable baseline to spot any microscopic differences. The data is now clean and organized. However, it still requires deep computational inspection to isolate the specific genetic anomalies that cause disease. This workflow flowchart shifts from mapped reads into step 4, variant calling. Here, tools like GATK hunt down single nucleotide variants, insertions, or deletions within the aligned data. Finding a structural typo is only half the process. Just knowing a genetic sequence is altered doesn't reveal what that typo actually does to a patient. That leads to step 5, variant annotation. Platforms like Anavar and VEP assign biological and functional context to the identified variants, mapping them to specific genes and regulatory regions. Step 6 involves functional impact prediction. We use tools like SIFT and PolyPhen2 to predict whether a specific mutation is benign or actively pathogenic, based on evolutionary conservation and protein structure. Finally, step 7 is visualization and reporting, where all this annotated data is compiled into interactive clinical reports. This sequential pipeline is the only reliable way to distill terabytes of raw noise into a singular, interpretable genomic report ready for an oncologist's review. To see how this precision oncology works in practice, let's look at a specific clinical mystery, the EGFR L858R mutation, frequently found in lung cancer. This Vano portal grid illustrates how the bioinformatics pipeline identifies this exact mutation. It highlights the genomic positions, transcript annotations, and variant effects, classifying the L858R mutation as pathogenic. Because the computational analysis flags this mutation and matches it against global cancer databases, oncologists know definitively that the tumor will respond to targeted therapies like erlotinib or afatinib. Matching a specific computational discovery directly to a targeted chemical cure is the core life-saving application of precision oncology. This circular diagram maps out how NGS applications extend far beyond cancer. This exact same pipeline was used to track SARS-CoV-2 variants in real time during the pandemic and continues to diagnose rare Mendelian disorders in newborns today. Hardware capabilities are also accelerating. Emerging technologies like single-cell sequencing and portable long-read sequencing devices, like the Oxford Nanopore system shown here, are pushing the pipeline's capabilities into the field. Human analysts can no longer keep up with the scale of this multi-omics data. Integrating artificial intelligence and machine learning is now an absolute necessity to automate these pipelines and reduce interpretation time. The NGS journey transforms a messy data deluge into the automated identification of targeted cures. Thanks for watching. Please like and subscribe to Computational Genome Biology.